All, I am Leena from Department of Information Technology. Today I am going to share about the digital image processing. So what is an image? So image may be a picture or a photograph or a visual data that is usually 2 or 3 dimensions. So what is digital images? An image which is in the discretized, that is defined on a discrete grid. So that is called as a digital image. It has a two-dimensional collection of the light values or a gray values. What is digital image? A representation of the original image by a discrete set of data points. Each of these data points is called as a picture elements, that is commonly referred as a pixels. Why digital image processing? Image is better than any other information form of human being to perceive. Humans are primarily visual creators. Above 90% of information about the world. A picture is better than the thousands of words. However, vision is not intuitive for a machine. Projection of the 3D words are the 2D images, that is a loss of information. So interpretations of dynamic scenes such as moving the camera and moving the objects. So what is digital image processing? Image understanding, image analysis and computer vision aim to imitate the process of human vision electronically. That is image acquisition, pre-processing, segmentations, representing and description, recognition and interpretation. So in the digital image processing, the important steps are the image acquisition, that is to be, the image is to be get from the source, and after getting the image is to be pre-processed, and the pre-processed image is to be segmented, that the region of interest, that is what information that is needed, that to be segmented, and that feature is to be extracted by using the feature extraction algorithms, and after we classify it the image. So this is the digital image. The digital image is equal to a multi-dimensional array of numbers, such as the intensity images are the vectors, such as the colored images. Each component in the image called as a pixels, that is associated with the pixel values. A single number in the case of intensity images, or a vector in the case of the color image. So the general procedure, that is the goal, is to obtain the similar effect that provided by the biological system. Two levels of approaches are to be used: low level image processing and the high level image processing. In the low level image processing, very little knowledge about the content or the semantics of the image is to be taken. In the high level image understanding, that is imitating the human cognition and ability to infer information contained in the images. In the low level image processing, very little knowledge about the content of the image is to be get. The data of the original image represent as the matrices of intensity values, that is sampling of a continuous field using a discrete grid. Focus of this course. The low level image processing, so this is the origin of the original image. So it is the human brain. So in the human brain, that it has a 3 into 3 neighborhood values. The pixel values are to be taken, that is a square, the square form that tells the pixel values. This is a pixel regions. So the low level image processing, the main thing that we are discussing is that image compression, the noise reduction, edge extraction. These are the pre-processing steps that to be done in the image processing. So the edge extraction, contrast enhancement, segmentation, thresholding, morphology, image restoration, image compressions. So all these things are to be taken by the pre-processing steps. So first, in the first image we are doing that image compression. In the second part of the image we are reducing the noise in the image, and the third we are extracting the edges, and the fourth we are contrast enhancements. We are enhancing the contrast of the image, and here we are segmenting the image. So the region that you are going to do the research, that part of the image is to be segmented, and we are limiting that threshold. We are setting some threshold values, and the last one, that is that morphological actions, the erection, what are the unwanted things that are to be that are to be deleted, and the image restoration, the original image is to be restored. These are done in the low level image processing. In the high level image understanding, it imitate the human cognition. According to the information contained in the image, the data represent knowledge about the image content and are often in the symbolic forms. The data representation is specific to the high level goal. So it is an element of the visual perception, how the human eye is getting that image. So this is the structure of the human eye. So if you are taking the human eye, its diameter is 2.0 mm, and the three membranes are enclosing the eye, that is the cornea and the sclera and choroid and the retina. The choroid contains the blood vessels of the eye, nutrition, and is healthily pigmented to reduce the extraction light enters and the wax colors. It is divided into the ciliary body and the iris diaphragm, which controls the amount of the light that enters the pupil. The lens is made up of the fibrous cells and are suspended by the fibers that attach to the ciliary body. It is slightly yellow and absorbs approximately 8% of the visible light spectrum, and the lens contains 60 to 70% of water and 60% of the fat. The retina. The retinal lines that enters the posterior portion, discrete line receptors are distributed over the surface of the retina. The cones and the rods they play a very important role. The cones are located in the fovea and are sensitive to the color. Each one is connected to its own nerve ended, and the cone vision is called photophic. There are three types of cones, that is the red, green and the blue. Next one is a rod. The rod are giving a general overall picture of the field of view. They see only the brightness, that is not the colors, and are not involved in the color visions. Several rods are connected to the single nerves and are sensitive to the low level of the illumination. The scopotophic are a dim vision or a night vision. So the applications of the digital image processing are in the field of medicines, defense, metrology, environmental science, manufacturing, surveillance and the crime investigations. So the applications in the medicines, they are using the CT scans, PET scans and mammograms. Many types of the scans, the digital image processing is to be used. So these are the various field where we are using. So what are difficulties we are facing when we are using the digital image processing? The human vision system tend to group the related region together, not odd mixture of the two alternatives. Attending to the different region or contours indicated a change of perception. This illustrates one more than vision in an active process that attempt to make the sense of incoming informations. So these are the key stages in the digital image processing, that the source of the image is to be called as the image acquisition. Next we have that image is to be enhanced and it is restored. Next the morphological processing are to be done in that image, and after that we are doing the segmentations. The object is to be recognized and the representation of description of the image, and the image is to be compressed and the color, the image processing. Thank you.